In every diploid cell, there are two sets of chromosomes, one from the maternal parent and the other from the paternal parent. And at the time of meiosis, that is the reductive division of the cell, these two chromosomes of the homologous pair, they separate from each other. But they do not separate as they are. Before separating into the daughter cells, that is the gametes, these chromosomes, they exchange some of their parts. And this exchange of chromosomal segments at the time of meiosis, it is supported by a proteinaceous complex, that is the synaptonemal complex. So in this video, we will be learning something about this synaptonemal complex that is formed during the meiosis. That is, it is the characteristic feature of a cell undergoing reductive division. Now, the prophase 1 of meiosis, that is the longest phase of meiotic cycle, and it is divisible into 5 substages: the leptotene, zygotene, pectine, diplotene, and dikinesis. Now, during this prophase 1, there's chiasma formation and crossing over. These are the facts that we are just revising. So, this prophase 1 stage, it is characterized by 4 main chromosomal processes. That is, within the chromosomes, 4 changes takes place during prophase 1. First thing is that the sister chromatids, they associate with each other. And when these DNA molecules, they are replicated during the S phase, the replicated DNA molecules, which take the form of sister chromatids in the chromosome, they do not separate from each other. We are not able to see these arms of the chromosome right from the replicative phase, because at that point of time there are certain proteins which connect these sister chromatids together. They are intermolecular glue. They join, they adhere these two different DNA molecules, that is the sister chromatids. And this cohesion is brought about by the protein cohesin. Second thing is that the chromosomal axis, it gradually starts condensing. The two sister chromatids, they are connected to each other, and in that very manner they start condensing. The nucleosome formation, the coiling, the supercoiling, gradually it takes place. And since the two sister chromatids, they are connected to each other and each chromosome is condensing, at this point of time, pairing of the homologous chromosome takes place. The third thing is that in between these homologous chromosomes, their pairing, it is facilitated by a proteinaceous complex, that is the synaptonemal complex. And fourth characteristic feature of this prophase 1 is that after the synapsis is established between these homologous chromosomes, crossing over takes place, a recombination takes place, the general recombination or the homologous recombination, about which we have learned in the previous video. Now, if we'll consider these various stages, so during leptotene, when we say thin thread-like stage, the homologous chromosomes, they come near each other and they start pairing. In the next stage, that is zygote, two threads. Here, this pairing of homologous chromosome is complete, and there is this synaptonemal complex. It establishes itself in the form of a zipper between these two homologous chromosomes. Then in pectine, this formation of this synaptonemal complex is complete, and at the advent of this diplotin phase of meiosis 1, prophase 1, disassembly of this synaptonemal complex, it initiates, it disassembles rather, and as a result of which we are able to see the bivalent form or the two sister chromatids, the chromosome shape with the two arms that becomes visible once this zipper opens. That is, this synaptonemal complex that started its formation during the leptotene zygotene was completed during the pectine. It starts disassembling. Now, if we look into the structure of this proteinaceous complex that is formed between these two homologous chromosomes. So this meiosis specific proteinaceous structure which pairs these homologous chromosomes at the time of synapsis, that results in synapsis rather, it is a tripartite structure. It has got three parts. There are two lateral elements which are connected to the homologous chromosomes, and the region, the approximately 100 nanometer region in between these two lateral elements is the central region. This central region is made up of two parts. Main thing is these elements, the transverse elements, these proteinaceous thread-like structures which join or connect these two lateral elements, the transverse elements. They are known as the transverse elements. And this central element, it provides support to these transverse elements. Apart from that, this central element, it has got the recombination nodules, certain proteinaceous complexes which are ellipsoidal. They are present at some distance from each other within this on this central element, within this central region. So this is the basic structure of a synaptonemal complex. Now, right from the very beginning, we are saying that synaptonemal complex is a proteinaceous complex. Many proteins, they are responsible, or rather they contribute to the formation of this zipper like, the ladder like structure that connects the two homologous chromosomes together so that crossing over may take place between them, or so that the homologous recombination, it may be facilitated between these two homologous chromosomes. So in this lateral element part, here green and blue region, it is made up of three different types of proteins. One, they are the cohesins. Cohesins, as we have said, they are the intermolecular glue. They connect two different molecules together, and these are the various types of cohesins that have been reported in the lateral elements of different organisms. Now, over here, I will like to mark it that this structure of synaptonemal complex, it is conservative within the eukaryotic organisms, starting from the yeast up to the human elements. And the what differs is the type of protein that forms this synaptonemal complex. The basic functioning, it is the same, but the sequence of amino acid, that is the protein, type of protein, it differs. Although these proteins which form this different parts or elements of the synaptonemal complex, they are orthologous. By the term orthologous, we know that orthologous proteins, they share a common ancestry, and during the time of evolution, certain changes, certain alterations, they take place in the genes. So the genes and the proteins with common ancestry, which diverse as the system evolves, they are referred to as the orthologous genes or the orthologous proteins. So these are the different cohesins that may be present in the lateral elements. Cohesins, they are present, but which type, that differs from organism to organism. The second type of proteins, they are the basic structural proteins, that is the synaptonemal complex protein 2 and the synaptonemal complex protein 3. Apart from this, they are the hormodomain protein. These proteins, they have been named for the first three proteins which were reported to have this particular structure, particular domain. And these three proteins which were first shown to possess this domain, they were the HOP1, the REV7 and the MAD2, MAD genes. So this name HOR, HO from HOP1, R from REV7 and MA from the MAD2. These hormodomain proteins, they are also different for different organisms, for different eukaryotic cells, but this lateral element, it comprises of the cohesins, the structural proteins and the hormodomain proteins. Similarly, these transverse elements which zip together these lateral elements, they are formed of proteins like ZIP1 or the synaptonemal complex protein 1 or the SYP1. Same thing applies over here also, different for different organisms, different types of eukaryotic cells. And these zip-like proteins, these transverse elements, they are basically dimers. So these dimers, they zip together these two lateral elements and form this synaptonemal complex. And the central element, it provides support to these transverse fibers or the transverse filaments of the synaptonemal complex. And see over here, they are the two sister chromatids of the homologous chromosomes, and they are joined together by this uh, element, by this synaptonemal complex, which is the proteinaceous complex. During the interphase stage, when the cell, it prepares itself for this cell division, at that point of time during the S phase of interphase, DNA replication, it takes place. Now this DNA that replicates itself results in the formation of two daughter DNA sequences or the two daughter DNA molecules. But at this point of the cell division, these two daughter DNAs, they do not separate from each other. The two chromatids that are formed during the S phase, they remain connected to each other by means of certain protein molecules that form the axial elements. And these proteins, they glue together these uh, daughter chromatids, and they are referred to as the cohesins. These elements, they are present between these two daughter chromatids, and at the time of prophase 1, when pairing between these homologous chromosomes they take place, at that point of time these axial elements, they take the form of lateral elements. They are approximately 50 nanometers broad, and now their function in the form of lateral elements will be to glue together the two homologous chromosomes. After that, these transverse filaments, they organize between these two lateral elements in the central region, which is 100 nanometer wide. These transverse filaments, they form a zipper-like structure connecting the two lateral elements of the two homologous chromosomes that come to lie parallel to each other during prophase 1, the leptotin stage. And this zipper-like structure, it is stabilized by another type of proteins, that is the central region proteins or the central elements, which stabilize this central region. At the point where crossing over takes place, now we have seen in another video that at the time of recombination of the homologous recombination, when crossing over takes place, certain protein assemblies, certain Recce type of homologous proteins, they organize and they facilitate this process of crossing over in this region. So they form special ellipsoidal molecules known as the recombination nodules, which are present in these central regions. This synaptonemal complex, it plays an important role, or rather it is responsible for gluing together of the homologous chromosomes, for the alignment of the homologous chromosomes, or for the synapsis of the homologous chromosome. And it facilitates, it makes easy the exchange of the parental information or exchange of the various chromosomal segments at the time of homologous recombination. And as we know that this homologous recombination is very essential or crucial for the generation of the variations during the meiotic division, and for the segregation of these homologous chromosomes of these various genes, the Mendelian segregation of alleles, that is brought about by this homologous recombination. And this entire process is facilitated by a proteinaceous complex, that is the synaptonemal complex. In this way, this synaptonemal complex, it stabilizes the pairing of the homologous chromosomes at the time of prophase 1. After that, it promotes the formation of crossover. It helps in the formation of the recombination nodules. And apart from these two functions, which results in the segregation of different genes between these homologous chromosomes, it also restricts the number of crossover that may take place between these two homologous chromosomes. So it is responsible for the crossover interference which is defined as the ability of the nascent crossovers to inhibit the formation of other crossovers in its vicinity. Because this proteinaceous zipper-like structure is formed, and these crossovers may take place only in the region of the recombination nodules, which are separated from each other along the length of this synaptonemal complex, which zips together the entire length of the homologous chromosomes. So this proteinaceous complex, it facilitates the various processes that take place during the prophase 1 of meiosis and are responsible for evolution, for the segregation of various genes, factors between different gametes that are produced during meiosis.